everyone. Thanks for joining us for our Energizing Human Physiology with Data Collection workshop. We will give it a few moments so people can join us here in our Zoom room. And while we're waiting, I wanted to go over a few housekeeping items. One is feel free to use the question and answer feature as well as the chat feature to ask your questions during the session. We want you to be able to have an opportunity to ask questions, and we'll be answering those throughout. We'll do our best to answer everyone's questions, but if we happen to not get to your question, we will follow up with you after today's webinar. I'll be sharing a link to a shared Google folder in our chat in a moment, and it has some additional resources that your presenter, John Melville, put together for you, including some sample data for today's experiment. And then I will also be sharing where you can find the recording of this webinar on our website in the chat towards the end of the session. And to kick us off while we're waiting for people to come in, I'm going to deploy a quick poll just to get a better idea of the direction for our webinar. And we're just curious if you could answer if you're here more for the spirometer side or if you are here to learn more about graphical analysis, or both. And John, it looks like we have most people on the spirometer side and then a couple on both.
Okay, great.
So thank you everyone for taking that poll. And the next part is just to go over the agenda. So you'll meet John in a moment. He'll talk a little bit more about himself. He'll be doing an introduction to the lung volumes and capacities experiment and talk more about that. And he'll also go over some different options for remote learning or hybrid classrooms and give you some options for those. And we'll be doing the hands-on demonstration. And again, feel free to ask questions. And so I'll pass it over to John.
Hi. So I just wanted to introduce myself really briefly. My name is John Melville. I'm the Director of Biology and Biology Staff Scientist at Vernier Software and Technology. I got my PhD in zoology at Oregon State University, and then I did a postdoc at University of Oregon. But most importantly, I'm a former educator and teacher. I was an associate professor of biology in the biology department at Warburg College, but I also taught briefly at Lane Community College as an adjunct. And I've taught lots of courses. I've taught majors and non-majors courses in biology, neuroscience, and a lot of courses also related to human physiology. And in almost all of my courses, the focus was on active learning and an emphasis on data collection and inquiry and having students do their own experiments. I also was one of the first people at Oregon State University as a grad student to incorporate data collection technology into the animal physiology courses that were there. So I just wanted to let you know that we're here for you. If you have any questions, ask them in the QA. Someone will be there, or Angie will be there sending them my way and asking me questions if there are any questions in chat. And if time allows, we'll discuss questions at the end of the talk. The other thing I want you to do is to find graphical analysis, download it if you do not already have it, and just make sure it's version 5.4. No sensors are required. If you go to our website, I'll just move out of the presentation here really quick. This is where it's located on our website. Can you all see that still? So here's Vernier graphical analysis. You can get that just by tapping on downloads and then going to graphical analysis. And you can get that for Windows or Mac here. If you have a Chrome device, you need to go to the Chrome Web Store. If you have a Google device, you need to go to the Google Play Store. And if you have an Apple device, iOS device, you need to go to the Apple Store. Great. What I wanted to do next was just, if you've done that, you don't have to, just make sure you get it right now. I'm going to continue on briefly with the presentation here. What I wanted to do is talk about the Vernier GoDirect spirometer. And what we're going to do is we're going to look at a classic lab experiment, which looks at lung volumes. And in this lab experiment, you basically get something that looks like this graph down below, where we're looking at tidal volumes, and then vital capacity, and then inspiratory reserve and expiratory reserve. And really the main thing that most people are interested in is this graph down below here, the volumes. And this lab is in one of our books. The latest version of it is in Human Physiology Experiments, Volume 2. So there are 2 versions of Human Physiology Experiments. Human Physiology Experiments, Volume 1 supports a smaller subset of sensors. Human Physiology Experiments, Volume 2 supports our blood pressure, GoDirect blood pressure sensor, and GoDirect spirometer, and has some more advanced labs in it. So for a complete like college level course or for an advanced human physiology course, you would want to get both books. So any questions so far before I move on to the GoDirect spirometer and the lab in question? Angie put a link to the Google Drive. And in that Google Drive, you will see this lab as written. There's the teacher's instructions that are there. I'm just in the Google Drive right now. The teacher's instructions, student instructions, and I even have sample data for you to use because we can load up the sample data and take a look at it. All right, so let me go back to the presentation. Just to let you know where we're going to go, Vernier software and technology is what we call for teachers and by teachers. So I'm a former educator. We don't have quote unquote sales reps. Everyone that works in the tech support group, for the most part, it is a former teacher in some way. I will do a live demo of Vernier Graphical Analysis Pro. No sensors are required. You don't need anything, but I will show that to you later. And then I'm going to go over the features of the GoDirect spirometer. And the GoDirect spirometer is designed so that you can compare and analyze respiratory patterns, measure tidal volume and other lung parameters, and also like analyze lung function. But it also has 2 very special features in it that our old spirometer does not. And that it has a special channel to control for baseline drift, and it has special channels for longer experiments. So if you're interested in that, please let me know and I'll go over some of those more advanced features. So any questions? Just feel free to put them in chat or the Q&A, and Angie can relay them to me. All right, so what I'm gonna start off with then is just doing some basic data collection and introducing you to GA. Graphical Analysis Pro is really great because it can be used with in-person learning that we have right up here, but it can also be used in a remote asynchronous environment, so you can insert video into it. And then we also have a remote synchronous learning style where we can data share data. So I can actually take data that I've collected and I can send it out to you remotely, and we'll demo that later. But let's just start with the live demo. So remember, I'm going to be using the GoDirect spirometer, and what we're really looking at are these features. I'm going to be breathing in and out, and we're going to be trying to look at tidal volumes, vital capacity, which is the greatest inspiratory volume that I have, and then the difference between inspiratory and expiratory volume, and then the difference between the tidal volume and the rest here for inspiratory is your inspiratory reserve, and then expiratory reserve, and there's some residual volume. And this is the most classic experiment that is done with this barometer is this lab. So let me just open up GA. Here's graphical analysis right here. Let me zoom up a little bit. Actually, let me just organize this a little bit. I'm just doing a new, I've just done new experiment here. So let me just do file, new experiment. Don't save. Normally when you launch graphical analysis, this dialog will come up and it'll ask you if you want to do sensor data collection, data shading, or manual entry. Now that's if you don't have something already attached. What I want to show you next here is the GoDirect spirometer itself. And some people get confused because they think that these are wireless sensors, which they are. The GoDirect spirometer does have Bluetooth in it. But if you look at it, at the bottom, it has this little USB cable or USB port. So I can take a USB cable, a micro USB, I can plug it directly into the spirometer, and then it will connect to GA. So let me just do file, new experiment. There we go. And as you can see, it's already set up now to collect data. It's the spirometer is attached. So, oh, go back there. Pardon me. So the spirometer is attached by USB. It's ready to record data. Here's, I'll just bring up the GA. Now, if we take a look at the spirometer again, it's really small and straightforward. You can see it says, don't put any water in here. This is the flow head. It has a little sign here that says inlet. That's the way where you're going to breathe through. You wouldn't want to put the filter on the back side. You would want to put it on the front where it says spirometer. Now remember that each student needs to use their own bacterial filter. So this is a bacterial filter. It goes on the spirometer, pop it on there like that. And then you breathe in and out and in and out. And this protects the spirometer from the student and the student from the spirometer. Once again, every student is required to have their own bacterial filter. What I like to say is every subject. Now, when I taught, I used to have students write their name on their bacterial filter, and then they could reuse it. They could put it in the bag because then they could breathe in and out. So you wouldn't have to throw out a bacterial filter every time you used it. But it still is important that it's one per student. The GoDirect spirometer also comes with these disposable mouthpieces. So those go on here, and you put the spirometer on there like that. So that would be ready to go. There is a company called AM Systems, which sells this thermoplastic blue mouthpiece. And I like this mouthpiece. If you're interested in getting a hold of it, I can tell you where to buy it. I like it because there's a few things that you need to be aware of when you're doing spirometry, and that's that, essentially we're going to be putting this whole system in our mouth or in my mouth. So if you switch over to the other camera, right, we're going to put this in my mouth and blow through it. It's also important that you don't blow through it like you're going to be blowing on a trumpet. You want to actually insert it into your mouth. If any of you have ever been a scuba diver, you would know that it's really hard to breathe normally when you have a large object like that in your mouth. So what typically happens is tidal volumes are just going to be larger than normal. Most people would assume that tidal volumes would be about half a liter. Like for me, doing this activity, doesn't matter how many times I relax, it's going to be more like 1 liter. All right, so that's the spirometer all set up, ready to go. I'll just do the actual experiment really briefly, which is just an inhalation, exhalation, 3 inhalations and exhalations. And then I'll breathe in as hard as I can and exhale as hard as I can, and then do some recovery breaths. We should be able to do the whole experiment very quickly. I'm going to move this camera out of the way so we can focus on GA. The other thing that I want to show you, I'm going to turn these on in the background and then we'll take a look at them later, is the default when the system sets up is to give you flow rate and volume. But if you click on that little dialog down below and then tap on sensor channels, there's a whole bunch of other channels that are there. So you can also look at differential pressure, you can look at adjusted volume, cycle volume, and you can even calculate respiration rate. I'm going to turn on adjusted volume for a minute. And then I'm going to tap on the y-axis here, and I'm just going to hide it for now. We'll come back and take a look at it. So now let's just do this experiment. So I'm going to breathe in and breathe out. Breathe in as hard as I can, breathe out as hard as I can, and then we'll analyze some data. All right, so that means I'm not going to talk for a while. All right. So essentially, that's the classic experiment, is breathing in, breathing out, breathing in as hard as you can, breathing out as hard as you can. In most cases, flow rate is actually not important. We don't actually analyze it. So I'm going to tap up here under the view options. So top right-hand corner. And I'm just going to say one graph for now. And then I'm going to tap on the y-axis and I'm going to pick volume. And I'm going to turn off flow rate. And essentially what we do in this lab is you then find each tidal volume. So if you see, if I go to the peak here, from the base to here, I started off at 0 and the peak here is about 0.8 liters. Right, there's a little bit of drift. So from 0.14, the next one I would subtract 1.05 from 0.14. So as long as you measure from peak to trough or trough to peak, like if I highlighted this whole region and then went to say view statistics, it's going to give me the delta Y, which is from the peak, from the highest point to the lowest point, which is the best way to analyze that. You can also get vital capacity by doing the same thing, going to this, the highest peak to the lowest peak, and then doing the same thing. View statistics. And you can see that's about 3.11 liters. Now, you can get rid of this baseline drift. It's not critical. I mean, as long as you are measuring, like I said, from peak to trough, across the entire volume, it should be fine. But you can turn on the adjusted volume channel. And what you will see is it does that for you. So it finds where each breath ends, or inhalation, exhalation, and then it re-zeros itself. So you can see here, that's about 0.8 liters. That's about 0.9 liters. This one was a little big at 1 liter. And, 2.27 minus 0.18. So you could still calculate that. And then it's going to readjust once again. And you can see that the recovery breaths are a little bit larger, 1 liter, 1.121. But the adjusted volume channel will not work unless you turn it on. But you don't have to have it on. Like to do this whole experiment and get it to work well, all you have to do is, the key things about using the spirometer are holding it upright and still. So holding it still, not having it tilted, not having it tilted sideways, but straight up and down, and then just trying to breathe through normally. Usually you'll wind up with problems if you lean back and breathe in, then lean forward and breathe in. You just position it where you're going to be using it, and then just have the students hold it and breathe in and breathe out. You should be able to get decent data. Questions about that? No questions?
No questions yet, John.
All right, so let me briefly show you. Well, we can come back to that. The other cool channels that are in here, what cycle volume and respiration rate do is they're designed so that if you were doing a really, really long experiment, let's say you were doing an experiment that was going to last minutes, what they do instead is they will just find this peak volume. They just find each peak and they plot it. So what you would see is just a series of points for each inhalation, exhalation. It takes that difference and then it'll plot it. And then over a period of time, it will also calculate the respiration rate. And that's a much better way to do long-term experiments because the spirometer collects 50 samples a second. That's a lot of data. So if you're going to be doing or trying to do a much longer experiment on the order of minutes, those channels are just much more useful. Great. So while we're here then, let's go back to the presentation really quickly. And then let's just briefly talk about data sharing. So what we can do is, here's the Q&A. Let me see what the question was. Yeah, yes, you could do it to do an exercise respiration rate lab if you wanted to. The real trick in that case would be making sure it sits still. But yeah, you could do exercise. You could look at the effect of exercise, etc., and then look at it. That's actually kind of what it was designed for, is so that if you were going to be looking at a person slowly increasing their activity or decreasing, you could actually see it. Just the trick would be, how do you hold it still while they're exercising? But yeah, you could do a respiration rate lab. The Go Direct respiration belt just might be better for a question like that, just because it's not so invasive. Having something in your mouth while you're exercising can be problematic. But yes, you could definitely do it. So let's just go back to data sharing. So I wanted to show you a really interesting way to share data. So that was in-person learning. But if you were the instructor, or maybe you had a group of students, pardon me, that were working from home, one of the things that you could do, pardon me, you could go up to this top corner here where it says data sharing. And if you select data sharing, you can actually start a session. And then what you should see is this little number here, SOT9MJ. So I'm just going to remember that, SOT9MJ. And then let's see, I'm going to have, let's see here, pardon me. So here is the teacher's view. And then I'm going to bring up a Chromebook that I have sitting over in the corner. There we go. And so this Chromebook is sitting off to the side. It's completely different from the data that's here. And then what I could do if I was the student is I could open up Launch Graphical Analysis, say New Experiment, and then I would actually select data sharing. And then it's going to ask for the source code. Well, what's the source code? Well, remember, if I look back up here, it's the source code, or the ID code, is SOT9MJ. So I'm going to enter SOT9MJ. Tap connect. There, and then now I have sent data remotely to this other computer, and you could do that yourself. And that data is live, and it can be analyzed. So I could turn on different channels, volume, I could look at flow rate, and I could analyze it. And that's all independent of the teacher's graph. So this is real data that has showed up in real time on that person's device. Now, if you want to do this, is anybody interested in doing this? Because you could do this actually right now. If you launch graphical analysis, I can show you how to enter the code that you need into your computer to get this to work, and you could see this data. Is anybody interested? If you look on your version of GA, let me just hit style, hit file. In normal graphical analysis, file, new experiment, don't save. If you look at the screen here for the Chromebook, there's this little TIE Fighter icon up here. Normally, if you do not have GA Pro, this is Graphical Analysis Pro, there is a shield here. And then what you would do is you would enter this, oh, hold on, where'd that go? You would enter this license code that's located down here below here, or a license code. And then you would be able to turn it into GA Pro. All right. So the last thing that I wanted to show you in GA Pro is, let's go back to the presentation. There are also videos and experiments that live in GA Pro. The spirometer experiment does not live in GA Pro, but this is actually a super nice and very interesting feature, so that if a student, you could make your own video, you could easily take a video of doing the spirometer experiment and put that into this lab. And let me just show you how that works. If I go to GA here, and I'm just going to do new experiment, I'm not going to save this data. If I go to see experiments, it's going to list all the experiments that are available. And here's a nice one, monitoring EKG. I can launch that. And now what I can do is if I go up here and I hit the enable replay button, I can actually show this entire experiment. So this is my friend Walter. I'm going to hit the collect button, the start the replay button. He is going to put some electrode tabs on himself. Oh, that's the wrong side. So he's going to put it on the right lead where it needs to go. Put the red lead there. There will be something that will tell us when data collection starts, and then there are the EKG traces coming in. So this is another really nice feature of GA Pro, that you can actually insert videos with data. So once again, there are 3 environments that you can use this one tool, GA Pro, in. You can do live data collection like you normally do in a classroom, collect data, analyze it. The GoDirect sensors are great for that. You can also use data sharing. So if you have students that are not in the classroom, but you have access to the internet, or maybe you have a hybrid classroom where some students are in the classroom and others aren't, you can use the data share feature to send that data to them. Or maybe they're just, working completely remotely, and then they're going to come back and tell you what they've done. You could actually record a video, insert it into GA Pro, and then share that with them. So there's lots of different options. Now for remote education, just in the last minute we have here, there's a whole set of data for remote education that lives on the website. If you go to our website and you hit Explore Remote Solutions, what you will be able to find here, let's just go to high school. Go to high school. We have an entire sample data library. So if I'm just scrolling down here, if I go to the Analyze Experiment Data at Home, I can view the free library. And if you browse it, I just wanted to show this to you, and then we'll take questions. These are all the experiments that are listed here with some sample data and even related video. And what's really amazing is that for Human Physiology Experiments Volume 1 and 2, we have all of the sample data for all the experiments. So if you want to just look at what the sample data should look like, and how it would work, all of that is freely available right now. And with that, let's just take some questions. Does anybody have any questions? And they could be questions about the Graphical Analysis Pro, they could be questions about the spirometer, anything at all.
I am not seeing questions come through yet. Oh, here's one that I can answer. Yes, the video will be, the recording of today's session will be available, and I'll put the link in the chat now.
One last thing is the bacterial filters are really effective. We did some research on them, and they're as effective as if you're looking for information about how effective they would be at filtering out viral particles, just ask. Just email biology@vernier.com and just say you have a question about the bacterial filter, and we'll answer it for you. The answer is yes, they're about as effective as I'm forgetting the name of the actual filtration efficiency, but as good as a clinical-based mask. They're actually quite good. But I can get you more information on that. You could do a lot of spirometry-like experiments using our GoDirect respiration belt as well, and then you wouldn't need to breathe through anything. And we can help you out with that as well. that's actually another really interesting product.
Well, thank you everyone for taking the time out of your busy schedule to join us. We appreciate that. I did put the link to where you can find the video recording of today's session, and feel free to reach out to John or anybody on the biology team. If you have questions, you can reach them at biology@vernier.com. Thank you.
Thank you.